Hi, my name is Tony Tillson, Head of Communications and Engagement at Cambridge University Health Partners, and I'd like to welcome you today to today's Cambridge Biomedical Campus on virtual tour event. Since we moved our talks online, we have had thousands of people logging on to hear from the best and the brightest clinicians and researchers and academics from across Cambridge. All of the talks are now available on our CBC YouTube channel. This lunchtime, I have the privilege of introducing James and Paul, leaders of the Ovarian Cancer Programme at the Cancer Research UK Cambridge Centre, to hear about the pioneering research that's aimed to improve outcomes for ovarian cancer patients. James is a senior group leader at the Cancer Research UK Cambridge Institute, Professor of Ovarian Cancer Medicine in the University of Cambridge Department of Oncology, and Honorary Consultant of Medical Oncology at Addenbrooke's Hospital. While Paul is Director of Teaching for the University of Cambridge's Department of Public Health and Primary Care, and Professor of Cancer Epidemiology in the Department of Oncology. As usual throughout the talk, if you are watching through Zoom, you can ask questions at any time using the Q&A box, which will be addressed at the end of the talk. We will then do our best to include any comments made by those on YouTube Live, but please be not too disappointed if yours is not included, as we often have a very chatty audience. And finally, to reassure you all, you won't be seen or heard throughout this event, but it will be recorded to be accessed on our website and the CBC YouTube channel in future. So without any further delay, let me hand over to Paul and James. Thank you.
Okay, thank you very much for that introduction. So I'm going to kick off by talking about the work that I've done around ovarian cancer for the past 20 years. And my group has been doing work into understanding inherited genetic susceptibility to ovarian cancer. And I'm going to kind of give you a lightning tour through the research we've done over the whole 20 years. So I'm not going to go into anything in great detail, but it's to try and demonstrate how research like this has a very long run-in before it leads to any kind of clinical impacts, but the clinical impacts are real. And I also really want to emphasize that this research is a real big team effort over many, many years. But before I start, I want to say a little bit of basic genetics, because all of the research that I'm going to talk about is research into genetics, and you need to have at least some understanding of the problem and the issues that we're talking about. So I'm sure most of you know that the human genome is made of DNA, and that DNA is simply a string of bases, or letters if you like, in a long chemical line, and there's 4 possible bases, A, C, G, and T, and it is the unique sequence of all those bases that make up the information contained in the human genome. And in total, there's about 3 billion bases in one human genome. So that's a lot of letters in a row. And just to put that in context, the King James Bible is about 3,100,000 letters. So the human genome is about 1,000 Bibles worth of information in terms of the letters involved. So all our cells, apart from red blood cells, and our cells like sperm and eggs, have 2 copies of the genome. And those copies are bound up in 23 pairs of chromosomes, so 1 copy in each of the 2 chromosomes. And so one set of chromosomes is 1 copy and the other set is the other copy. And we inherit 1 set from one parent and the other set from our other parent. So human genomes are not all alike. In fact, each genome is unique. And as such, there's no such thing as the human genome. When we talk about sequencing the human genome, and the human genome sequence doesn't mean any single sequence, but in fact, a sort of average across all humans. And in fact, a human genome, if you take 2 random human genomes, they're about 99.9% identical. So they vary at about 1 in 1,000 bases. So that means if you take two such genomes, about 3 million of the bases are different from one another. And to put that in some context as well, if we were to compare a human genome with a chimpanzee genome, they vary at about 1% of bases. So about 30 million of the base would be different, which means most of them are in fact the same. And genetically, we are quite closely related to other apes, including chimpanzees. So genetic variation is where one genome varies in comparison to another. And so we can characterize that in several different ways. There's the type of variant that occurs, how common that variant is in the population, and then what my research is focused on is understanding how that variation may affect a person's phenotype. So a phenotype is, what we are. It could be something like human characteristics like height or skin color. It can be characteristics like blood pressure. It can be anything that we can measure in a person, or for me, the phenotype of interest is cancer risk. So just to explain what types of variant there are, and there's many, many types. I can't go through all of them in great detail, but this line of letters here just represents a bit of DNA of about a dozen bases or letters. And if we call that a reference genome, then at this position here that I've highlighted with a ring in C, the G has been changed to a C. And that is a typical single variant change in the human genome. Other variants, sometimes you can lose one base. So in this sequence, where the circle is highlighted, the G has been lost. And so that little bit of sequence is one base pair shorter than the reference. And the opposite of that is you can actually gain an extra base. And here we've gained an extra C as well as the G. And so they are examples of single variants, but this can also occur where you can gain or lose or change lots of bases in a row. And indeed, you can gain and lose thousands of base pairs in a row with very large changes. So any given variant can vary in frequency from being extremely rare, indeed unique to one human, or they can be quite common and be present in a large proportion of genomes. And so there are about 15 million different variants across the genome that occur in 1% or more of all human genomes. And there's 50 million variants that occur in more than 1 in 10,000 genomes. So there's a lot of variation in the human genome. So understanding the effect of this variation on our phenotypes is what genetic epidemiology is all about. So the first thing to say is lots and lots of variation has no discernible consequences, and the variants don't seem to have any meaningful effect on phenotype, at least. On the other hand, very small changes can have huge effects. So for example, a single base out of 3 billion bases can make a major difference to phenotype. So a common cause of dwarfism, which is the achondroplastic dwarfism, is caused by a single change in the gene FGFR3. And that obviously has a major effect on an individual's phenotype. But on the other hand, extremely large changes, loss or a gain of thousands of bases, may have no discernible effect at all. Of course, there's lots and lots of ways that a variant can affect function at the molecular level, and I'm not going to talk about that. But the important thing to understand is that a lot of normal human phenotypic variation, why you and I differ from each other in all sorts of ways, are due to the common variants in the genome. And so my work has all been about understanding the science between genetic variation and the inherited basis of breast and ovarian cancer. And I'm going to run you through some of the things that we've done over the last 20 years. So if you are hoping that this was all going to be very simple, I'm very sorry, but this cartoon kind of says it all. I wanted to mention one other thing, because I know James is going to be talking a little bit about somatic genetic variation and cancer as a somatic genetic disease, and I'll explain that in a moment. So, as I said at the beginning, all our normal cells, which are at the top right of this figure, so each one of those little circle represents a single cell in the human body, they are, to all intents and purposes, identical genetically to start with. But every time a cell divides, it has the potential to gain a mutation. And so while most of our cells are the same, every now and then a cell will get a mutation when the cell divides, and so it will differ from all the other cells in our body. And that's what we mean by somatic variation. It's not present in all the cells, it's only present in some of them. And it is in fact somatic variation that is the cause of most cancer. All cancers have abnormal genomes, but most of those abnormalities aren't inherited, they occur during life. What I'm interested in is how the genetic variation between individuals in the normal cells affect this process of going from normal to cancer. So we know that the variants we inherit from our parents might be associated with the risk of developing cancer. Lots of data to tell us this, but broadly speaking, people who have a close relative who developed cancer, they are at increased risk of developing the same cancer. So if you have a relative developed type of cancer X, you're at slightly increased risk. And for example, somebody with a first-degree relative, that's a sibling, a parent, or an offspring with cancer, they're at approximately twice the risk of getting the same cancer as their relative compared to somebody without such a family history. And we know from twin studies that most of that increased risk is due to shared genetics. And this sort of familial risk of cancer has been known for a long time. This is a scan of a book published in 1866. And you won't be able to read it, but on this page of the book, there's a multi-generation family described in which many of the women had cancer of the breast or cancer of the liver. And this is almost certainly a family caused by a mutation in the gene BRCA1 or the gene BRCA2. Obviously, when this was published, the genetics, human genetics, was not well understood. So as I said, there's lots of different ways of characterizing genetic variation. So the frequency is very important. So we know that rare variants can confer high risk of cancer. They could also confer moderate or even low risks of cancer. But more common variants that we might think of as not rare, not common, but uncommon, they can't be associated with a high risk of cancer because that would mean a lot more people would be getting cancer than they do, but they might be associated with a moderate or even a low risk. And then finally, common variants can't be associated with moderate risks or high risks because there would be a lot more cancer if they were, but can be associated with low risks of cancer. And so my work over the last 20 years has really been looking at first high risk variants, then low risk variants, then finally moderate risk variants. And so really the study of high risk variants predates the research that I've been involved in and started in the 1990s with linkage studies in large families with many affected individuals. And these studies identified the genes BRCA1 and BRCA2. And we now know that rare variants in these genes are associated with a high risk of breast and ovarian cancer and some other cancers. I should point out that the individual variants are very rare, but in combination, about 1 in 3 individuals in the UK would inherit a faulty variant in BRCA1 and BRCA2. And I started my PhD in the 1990s studying BRCA1 and 2. And we initially started off by setting up a study which was called SEARCH, in which we invited individuals with cancer to provide a blood sample for DNA and to fill in a questionnaire. And this was to provide a resource for genetic epidemiology studies. SEARCH ran for 20 years in terms of recruitment. And you can see from the numbers here that we've recruited many, many thousands of individuals with breast, ovarian, colon, prostate, and womb cancer. I'm going to say a little bit about my work on ovarian cancer since we first started with search. So some of the research that we've done, firstly, characterizing the contribution of faulty variants in BRCA1 and BRCA2 to the prevalence of cancer, understanding the risks associated with faulty versions of BRCA1 and BRCA2. Then later on, we started looking for common variants associated with cancer risk, then looking for uncommon variants associated with risk. And over the past 20 years, we've published over 400 papers that have used the resources from search. So just some examples of this work. Nearly 20 years ago now, my colleague Antonis Antoniou and I, together with my then boss Bruce Ponder and Professor Doug Easton, published one of the first papers of the risks associated with BRCA1 and BRCA2 for both breast and ovarian cancer. And this is a graph from that paper showing the risks of ovarian cancer over a lifetime. So if you take a 20-year-old woman over a lifetime, by the age of 80, she has about a 1.5% chance of getting ovarian cancer in the population. But somebody with a BRCA2 mutation, that risk goes up to nearly 20%. And for somebody with a BRCA1 mutation, the lifetime risk of ovarian cancer is over 50%. More recently, 10 years ago, we had got to the stage where the technology allowed us to sequence a large number of the search ovarian cancer cases to work out what proportion of cases were due to BRCA1 and 2. And we showed that around 10% of all cases of ovarian cancer are due to mutations in BRCA1 and 2. And that increases to about 15% if we consider the particular variant of ovarian cancer called high-grade serous ovarian cancer. You'll hear a little bit more about that from James in due course. And these two genes, BRCA1 and 2, explain about 40% of the genetic component of ovarian cancer. And then in the mid 2000s, we started looking for common variants using a case control study design where we compare the frequency of the variant in cases and controls. We formed the Ovarian Cancer Association Consortium in 2005 to do this as an international collaboration. These are the investigators at the first Ovarian Cancer Association Consortium meeting held in Cambridge in 2005. We did a lot of studies which were unsuccessful in identifying anything of any interest, which is why we were all feeling very sad for ourselves in the mid-2000s. And then along came the era of genome-wide association studies. The technology enabled us to do genotyping of hundreds of thousands of variants across the genome and tens of thousands of cases of controls. We did larger and larger studies over the next 10 years. And by the end of the last decade, we'd identified 37 common variants associated with ovarian cancer. And this graph just shows the progress over those 10 years. The blue line showing the number of variants identified, with 1 in 2009. Each of these little jumps is another publication with a few more variants. And then the red line is showing what component of the familial risk was explained by those variants. So we got up to about 37 variants, explaining about 5% of the genetic component. So to add to that diagram, of BRCA1 and BRCA2 explaining about a fifth each, with common alleles explaining about a 20th. We then moved on to looking for uncommon risk variants. So we couldn't do this using genome-wide association studies, but the sequencing technology had become cheap enough for us to be able to sequence lots of genes and thousands of cases of controls. And we published a series of these studies over the past decade and identified several genes, BRIP1, RAD51C, RAD51D, and PALB2 as genes that carry uncommon variants associated with an increased risk of disease. And we did all the characterization of those variants in terms of understanding the risks associated with the variants and their contribution to ovarian cancer in the population. I can't go through all of that in any great detail, but just one example is our estimate of the risk of ovarian cancer in a woman with a faulty variant in RAD51C or D. So here is the population risk of about 1.5% that I mentioned earlier. And then our estimate of the risk for a RAD51C or D carrier was that it's something over 10%. These dashed lines are confidence intervals on the estimate, which, as you can see, are really rather wide. There's more work to be done on these genes. So these rare or uncommon variants in these genes account for about 2% of the genetic component of disease. So there's still more than 50% of the genetic component of the disease we've not yet explained. And that's what our future work is attempting to do. It's attempting to find what explains this bit of our pie. So does this have any clinical relevance? Well, there are established interventions for women at high risk of ovarian cancer, and preventive surgery, the removal of the fallopian tubes and the ovaries, is very effective at reducing the risk of ovarian cancer and is offered to women usually after they've completed their families, often after they've gone through menopause, as a way of reducing their risk if it's high enough. We could have a long debate about what high enough is, but that's for another time. There are also therapeutic implications, and James may talk about this, in that if you develop ovarian cancer and you have a mutation in BRCA1 or BRCA2, there are now drugs that are specifically targeting those mutations called PARP inhibitors, and those drugs have come along in the last decade or so. So how have we put all this together? So my colleague Antonis Antonio, while we've been doing all this finding of the genes, has been developing comprehensive risk models that incorporate all the information that we know from family history and all the genes we've identified and lifestyle factors, and developed web-based tools that doctors and patients can use to estimate risk and help women decide whether or not preventive surgery is right for them. And that tool is now called CanRisk. It's used by thousands, tens of thousands of people from around the world. It's freely available on the web. That's the front page of the current version of CanRisk. And this is a tool that has been 15 or 20 years in the making. So that is a very quick run through of the work we've been doing in ovarian cancer over 2 decades. There's been steady progress, and it's starting to have real clinical impacts. And of course, there's an awful lot of people to thank in all of this. So that photograph I showed earlier was the first OCAC meeting. This was the last OCAC meeting. You can see how much bigger OCAC has got before COVID. This is we met in London a few years ago. And of course, we have to thank the people who funded this. We've had lots of funding from around the world, Cancer Research UK in the UK particularly, but also the US National Cancer Institute and the European Union have contributed lots of money to enable this work to go ahead. And I think we're leaving questions to the end, and I'm going to hand directly over to James. And I don't know how to get back the stop share. There we are. James, all yours.
Thank you very much. Let me just share my screen. Can I just check that everyone can see a slide that says, how can we personalize treatment for ovarian cancer?
Yes, we can.
Fantastic. Well, thank you for those kind introductions, and also thank you for Paul for getting over the principles of how faults in inherited material, that of course are our genes, cause ovarian cancer. So what I want to deal with in the next few minutes is just to give you a sense of what the challenges are around treating ovarian cancer and trying to choose the right treatment for women with ovarian cancer. So without more ado, let me just check this is working. So hopefully there should be a little red pointer on the screen. And the aims of this next few minutes are at a fairly high level to get over some really important points. Why is ovarian cancer difficult to diagnose early, and I'm going to explain why it's got quotes around ovarian in a second. Why is it difficult to choose or personalize treatment to patients? You know, how can we really do a better job of just giving individuals the treatment that will work best for them? And then at a practical level, what's our vision and how are we taking this forward in Cambridge University Hospitals Foundation Trust, or Addenbrooke's, where most of our patients are looked after? So the first thing to say is that the word ovarian cancer is a little bit of a misnomer, because the commonest type of ovarian cancer and the most serious type of ovarian cancer is this entity here called high-grade serous ovarian carcinoma. But in reality, this doesn't arise from the ovary, it arises from the fallopian tube. So this cartoon is showing the ovary in yellow and the fallopian tube in this pinkish color. And what's not shown here is the uterus. And of course, the main purpose of the ovary is to store the germ cells, and at ovulation, fertilization occurs in the fallopian tube and the developing embryo is carried down to the uterus for implantation and the development of the fetus and obviously human offspring. Actually, what we know now is that the vast majority, and I would say all cases of high-grade serous ovarian carcinoma, actually arise from genetic damage and changes to the lining cells of the fallopian tube. And secondly, this leads to several key features of the disease that are important in understanding it. So firstly, this, out of all the cancers that humans get, is exquisitely defined by mutations or faults that are acquired in cells, normal cells of the fallopian tube, in a gene called p53 or TP53. And this is an important gatekeeper for keeping cells, stopping cells developing cancer. And in 97% of patients, this gene is lost or damaged. A consequence of that is that, as I'll come back to in a moment, high-grade serous ovarian carcinoma is one of the most complex genetic cancers in terms of its genetic change within cancer cells. And I'll show you some pictures to explain that. And overall, we haven't changed survival for this cancer over the past 20 years. I think there are strong grounds for thinking that will change, at least for some of our patients. But in broad terms, although we have treatments that are often effective early on, we're not curing people. And we'll talk about that in the next few slides. And then the other really important thing to hang on to is, although this is a very complex disease, and although there's sort of chaotic patterns in the genes in these cancer cells, those are telling us that actually fundamental processes, particularly around DNA repair, are not working well in these tumors. But that could also be a vulnerability for the cancer as well as what's causing the cancer. And we need to exploit that. So mutational processes, or whatever that ongoing damage is, could be a marker for how to treat patients better. So the first thing to say is that many of you will know that ovarian cancer, many patients with high-grade serous ovarian carcinoma present with high-stage disease. In other words, a lot of disease around the tummy, the abdomen, that spread well outside the pelvis where the fallopian tube and ovaries are. And that's because of very easy spread from the tube. So just looking at this picture here, these finger-like projections on the fallopian tube are shown in this cartoon here. And what these cartoons are showing is that as you go from normal cells here to cancer cells that start to invade the tube but also shed, what happens is they can very easily spread around the abdomen because there's no physical barrier to spread. The fallopian tube is an open-ended structure. So that means that even before a woman has any symptoms of her disease, there are likely many implants of cancer cells on the surface of the bowel, on the surface of the liver, and on the lining of the tummy called the peritoneum. That means that when a patient is diagnosed, we say they have stage 3 or 4 disease. Stage is really a measure of how far the disease has traveled. But the other important thing to say about ovarian cancer patients is they have a lot of disease. In other words, very bulky disease and the collection of abnormal fluid. And this is a scan, a section of a CT scan from a patient. This is the vertebral column. This is the patient's left. This is the patient's right. And what we're seeing here is a complex pattern between normal air spaces. This is the bowel, but a lot of soft tissue relating to the cancer, shown in this light gray appearance. And in this patient had chemotherapy And actually, after a relatively short period of time, between November and January, in other words, 3 cycles of chemotherapy, the cancer has largely reduced. So again, you're seeing the vertebral column. This is the left, this is the right. You can see the bowel much more clearly now. You can see contrasts which the patient's drunk for this study. This is the white material, but there's much less of the cancer tissue. And the reason why this patient has responded so well is because she has a faulty BRCA1 gene, which is inherited, which makes her cancer more susceptible to treatment. And I'll come back to that in a few more slides. The other problem, though, with this very complex, chaotic genome is that it can evolve during treatment. And so I want to explain this with this story about this lady here, who in August 2010 had recurrence of her cancer. So she developed ascites, or fluid in the tummy, because her cancer had grown through treatment. And we were able to look at that and look at faults in the DNA in that cancer. And we found a fault in a gene called NF1. Now, that allowed us to go back and say, well, this gene might actually be a change since starting treatment. How many cells in that patient's cancer at diagnosis had this faulty change? So what I'm showing here on the left-hand side is what we knew at diagnosis from the tissues taken from this lady, from the ovary and the omentum. These are biopsies before chemotherapy started. And this is from her surgery in January 2009. And what the bar plots are showing is what's the proportion in each of these tissue samples of the cells with this NF1 mutation. And what this is really showing is that her cancer was a mixture of cells, both with this gene change, shown in blue, and cells that didn't have that gene change, shown in white. But what happened was there was selection for this gene change over time, and actually this is associated with worse outcome and worse response to treatment. So if we look at this in the cartoon below, this blue cell here has got the NF1 mutation, and as treatment goes on, it becomes enriched across the tumor and actually becomes the predominant clone at relapse, indicating that this worse outcome tumor cell, this clone, was the cause for her recurrence. So that takes us to the first problem I want to address. I'm going to talk about 3 problems in the next 10 minutes or so. The first is, how do we understand at diagnosis this very complex wiring diagram we will call the genomic profile of a patient's cancer tissue? And how can we do that in the clinic? So the reason why this is important, as I've touched on earlier on, is that our genomes are a wiring diagram and an instruction set for how the cancer behaves. And that has several important effects in the clinic. So the first, shown in this cartoon here, is how well the cancer can evolve. So just going back a moment, I was explaining how even a very single cells or very small numbers of cells at diagnosis, 3% here, probably 1.2% in the original cancer where it had developed, can grow into the major cancer mass that kills the patient. So that's indicated by this cartoon here. So if cancers don't change over time, their makeup stays the same, shown in blue. But if they can develop new variants or new phenotypes, going back to Paul's talk, this is similar to genetics. These cancer cells have got different properties, then they can evade treatment more easily. And this is defined by whatever that wiring diagram is. Secondly, the patterns of spread and where cancers spread to in the body can also be determined by genomes. And then lastly, what the ecology of that cancer is. Now, I'm using the word ecology here to mean the environment. And it's important to say that cancer is not just about cancer cells, it's also about the other cells that are around the cancer cell, cells from our immune system that are perhaps trying to kill off the cancer, but may not be doing a very good job of that, and other cells that may support the cancer. So these are shown in different colors here. The details don't matter, but the pattern and the instructions between what's happening in the cancer cell and its surrounding environment are very important. So Paul has talked to you about faults or mutations in genes. I just want to emphasize how extraordinarily complex those are in patient cancer samples. So the first thing to say is we inherit genetic material via chromosomes, shown in this cartoon here. But what can happen, and what does happen extremely frequently in ovarian cancer, is you get all sorts of cut and paste and deletion and gains of this genetic material at the level of the chromosome as well as at the level of the DNA. So think of this as the high-level view. This is how all the DNA is packaged up. And even in the packaging, it can be completely disorganized. And that's shown in this picture here. So on the top is a picture of all the chromosomes, these things here, in normal cells from an individual. So we have 22 pairs of chromosomes plus 2 sex chromosomes, either 2 X's in a female or an X and a Y in a male. And these chromosomes are shown in colors here. If we look at a cancer cell, for instance, from ovarian cancer, you can see, first of all, there are numerical abnormalities. Instead of just having 2 copies of chromosome 8, there's up to 5 copies of chromosome 8, so that's really not right. And then these multiple colors on the different chromosomes indicate how bits of one chromosome have been incorrectly and have been damaged, have fallen off one chromosome and then become stuck to another. And those changes, of course, change the way that genes are regulated. They stop genes functioning and make new abnormal genes by sticking together genes that should never be close together. So we can detect some of these changes by sequencing tumors from patients, and we've been doing this in the clinic for a while. And with this cartoon, I want to show you what that looks like in simple terms. So we have the chromosomes all the way from 1 to X. These are females with ovarian cancer, so they only have X chromosomes. And the chromosome 1 is larger than chromosome 22, so the gaps between here are slightly wider at the start. And then in normal cells, we have 2 copies of every gene, so we just have a horizontal line. But in a cancer cell, we have wildly abnormal copies, as I showed you on the previous figure here, very abnormal copies. But also we have changes in the amount of genes and very complex changes within them. So if we took patients and we look at their profiles, this is 117 patients from a national study we led. What you should see, if they were all normal, you just see a horizontal blue line. And what this movie is showing is how different all those changes are between patients. Every one of these dots is an abnormal number of genes, or it's an abnormal break in DNA. And this is a very difficult thing to read out. So how do we make sense of that when perhaps we're looking at the consequences of some important changes in the genes in a cancer, and we want to know the cause? So, and the other point, just to emphasize for a second, is that loss of genes like BRCA1 and BRCA2 can cause this sort of complexity at the cancer cell level in the cancer, but may actually be associated with better outcomes. So I'm only going to show one of these sort of statistical graphs. This is a graph showing the current results of a trial in which a medicine called a PARP inhibitor, so these are medicines, in this case, olaparib, but other medicines such as niraparib or rucaparib, are given to patients with ovarian cancer after initial chemotherapy and surgery. And in this really important trial run by AstraZeneca, patients either had a placebo treatment, shown in blue, or olaparib, shown in red, taken for up to 2 years after their initial treatment. And this is a survival curve. So at the start of the trial, at the start of treatment, all patients are alive on therapy. But as time goes from start of treatment, patients die, and that's indicated by, or progress, and that's indicated by the changes in the curve. So what the standout result from this is that obviously if you don't have PARP therapy, you do much worse. So by what we say the median survival, patients who didn't have a PARP medicine is around 12 to 14 months before their disease progressed. Whereas patients in red who received a PARP inhibitor out to 5 to 6 years after treatment, or about 44% of those patients have not progressed and are still alive without any worsening of their cancer. So this difference here is potentially the new numbers of patients who are cured by taking a PARP medicine after treatment. Now, this effect seems to be most important in women who've inherited a faulty BRCA1 or BRCA2 gene. But it's a marker of, if we understand the patterns and the damage, and we find treatments that target that, we may make a very big difference to survival, in addition to chemotherapy and surgery. And our approach in Cambridge to solving this problem is to try and look at the patterns and turn those into ways of classifying the disease for patients. So I showed you a few moments ago that movie of dots above and below the line. Those are called copy number profiles, and they indicate the numbers of gene changes in a patient's tumor. What we've done is, instead of looking at how many of those changes occur, we've looked at the type of changes that are occurring at the genome level. So we've classified those into 5 or 6 different groups and looked in every patient across their entire genome to say, what's the signature that's actually happening in terms of the patterns of genetic damage? And we've used machine learning methods to actually extract from that chaos 7 patterns that are associated with different mutational processes. And 2 of those patterns are associated with the faulty BRCA1 or BRCA2 gene. And importantly, these patterns start to indicate what treatment a patient might need, but also patients who may not respond to our existing treatments. So how do we get these tests into the clinic? Well, it's important to say that there are already some tests in the clinic that we're starting to use. We routinely do something called a myriad MyChoice test for everyone in the clinic, and this is a test, again, using next generation sequencing, but it looks at 3 features of the cancer tissues. So whether there's something called loss of heterozygosity, so have you lost a region of your chromosomes? Have you lost the ends of your chromosomes, or do you have other complex changes? And that test is an NHS test that is then used to choose whether patients have particular combinations of PARP therapy. So that's an advance that's taken us forward in the last 2 years to a new way of personalizing treatment. But we have to go further than that, because this is really an on-off test. And it's probably not complex enough to understand everything we're trying to do. The second problem is that not all patients get those tests because their tissue samples are not good enough for the DNA testing to be done. And so one of the projects that's been led between Birmingham and Cambridge by 2 very talented younger clinicians, Elaine and Gabrielle. Elaine is a gynae-oncology surgeon from Birmingham. Gabrielle is a medical oncologist in Cambridge, are really changing the way that we do this biopsy pathway. So in other words, a biopsy is a sample of tissue taken from a patient to make the diagnosis, with the strong aim of trying to get it right first time. That's what these initials are here, to improve the accessibility and the quality of tissue so that all women with ovarian cancer can get the best possible tests. The second advance that we've had in Cambridge is we've been working with Genomics England and NHSE and what's called the East Genomics Hub, which is the NHS laboratories that are doing these tests to bring detailed sequencing to patients in the clinic. And that's called whole genome sequencing. And as of now, we've sequenced about 20 patients with whole genomes that allow us to extract much more information and identify the patterns I've talked to you earlier on. But there's one last challenge I want to talk about in literally the last minute. That is, how do we bring all that information together in the clinic so that people can make decisions? And we have an electronic health record system in Cambridge, which really helps. We've moved away from paper notes, we're using electronic records, but we still need better ways of being able to present and analyze complex data. And in a way, what I'm talking about is we need to move between different scales of information about an individual patient. Everything from the macroscopic or relatively low resolution state, so that's about the imaging of patients and their CT imaging, that those ecological factors and the pathology information, so what proteins and what cells are present in a tumor, down to the DNA level and the RNA level. So that's a scale problem that we need to bring complex data sets together. And the area we're really trying to change is something called neoadjuvant therapy. So neoadjuvant therapy is the commonest way that women with ovarian cancer are now treated within the UK. And it means that a tissue diagnosis is made. That comes back to the project, the demo project that Gabrielle and Elaine are working on to improve. We make a decision by bringing all the data together on that patient. And then patients have chemotherapy before having surgery. Now, there's lots of good things about this process. It means that we can see if the treatment is helping the patient. It often makes their surgery safer and shorter because the cancer responds. But there's a number of problems. So we are still using simple measures to measure response. So simple measurements of tumor size. And actually, we don't have very good prediction tools. So it's still difficult for me to say to someone in the clinic when I'm starting chemotherapy, how much is your chance of responding to this treatment? And the good news is actually most patients do respond. So if you look at what happens, only a very small number, 6% of patients, their cancer gets worse despite chemotherapy. And about 61% of patients, even with advanced disease, have a complete or partial response. Mostly, in other words, these are technical terms, but have some response that shrinks down their tumor. The problem is about 33% of patients have no obvious benefit. They don't get worse, they don't get better, but they do have the downsides of having chemotherapy. And this group we need to understand much better and to be able to offer better treatments to. So the way we want to do that is by combining the genomic information I've talked about, radiomic information, that's detailed analysis of imaging tests, and also protein information from pathology tests. So I'm just skipping through this because of time, but in simple terms, that's being able to do a lot more about the CT data in terms of how much cancer there is, changes in the cancer tissues that we can see on radiology, which might be predictors of response, and coupling that with better presentation information and then detailed information from genomics and pathology. And we've just started, or about to start, a major collaboration with GE Healthcare to bring those pieces of data together in new dashboards that help clinicians make much smarter decisions about patients and to bring that data together. So this picture here is an example of a conceptual patient, obviously not a real patient, but the sort of information we'll be able to see at on a single screen and be able to drill down to particular advice that can be given about a patient's genome or their radiology. In this case, we're just clicking on how their cancer marker has gone with previous lines of treatment as a guide to how well they're responding. So this is going to be very important because without bringing the information to the fingertips of people in our clinical meetings, we will not make a difference. We will not drive trials forward. So I'm going to leave it there and stop talking. I just want to remind you of a couple of important conclusions. So firstly, there are real challenges in diagnosing and treating ovarian cancer. And by using the techniques I'm talking about, we can help improve that and reduce variability between hospitals, which is a problem I haven't spoken about in detail today. But to do that, we really need better molecular testing, and we need to combine this information in new ways that people can quickly make decisions and not miss out on important pieces of information. I'd like to thank our patient group and all our patients. So these are some of the patients from our patient group. They've been very important in helping us make these changes, particularly around sequencing, doing the studies and doing the trials. Our funders, particularly Cancer Research UK, who have done a lot of the funding for this, both across the centre and in the Cambridge Institute, and also other collaborators, particularly Mary-Lynn Alcaraz, who leads our ovarian cancer programme as the programme manager, and our wider clinical colleagues and members of the ovarian programme. Thank you very much.
Lovely. Thank you both, James and Paul. That was fascinating to learn more about an issue that I don't think enough people are fully aware of, kind of some of the difficulties and the challenges that you're helping to address. We've had a number of questions that have come in pre the talk and during the talk. So if I just ask away in the 10 minutes we've got, and then we can hopefully answer those as we go through. So I guess some of them have already been covered in what you've been saying, but someone's asked, how will the plans for the Cancer Research Hospital that's looking to be built on the Cambridge Biomedical campus, how would that help you in your work?
So shall I kick off with that? So I think there's several things that the Cancer Research Hospital is going to make a difference for. So firstly, all the research I'm talking about now and this collaboration with GE Healthcare will feed into the systems and the way we work in the Cancer Research Hospital. So we will be very patient-centric. It's not that we aren't patient-centric now, but we will be very patient-centric in terms of integrating in these complex pieces of information. Cambridge is the first hospital in the country to start doing whole genome sequencing in the routine care of patients. And we are going to build on that. That's happening now. But we're going to build on that with new algorithms. That's new methods of analyzing imaging data, the genomic data, to drive trials forward. And I think having an environment like the Cancer Research Hospital will make some of those steps easier, particularly around getting complex imaging or very specific tissue biopsies from patients to make the best decision for them. Secondly, I think it's really important that the Cancer Research Hospital is not just about patients in Cambridge. What we're really seeking to do is to use the learning and the research in Cambridge through the university, the Cambridge Centre, and be able to share that with colleagues across the Eastern region and across the country. And I think systems like the GE Healthcare system that I briefly described help us to scale out so that understanding of, say, the wiring diagram, the genome, can be shared virtually through video conferencing, as we're doing now, with colleagues that are looking after patients elsewhere. And similarly, the machine learning techniques and the new algorithms for analysis of imaging data can be shared in a way that really upscales everyone's care and means postcodes are less of a determination about whether you get the best options. Thank you, but that was a great question.
Paul, was there anything you wanted to add to that at all?
No, I don't think so. I mean, I think that the Cancer Hospital won't directly affect the research that I mainly do, which is population-based research. You know, we don't use the hospital, for example, to recruit patients into studies like search. Of course, just the general environment that the Cancer Hospital will produce in terms of promoting cancer research in general will be a good thing, but it won't be a direct effect on most of the work that I do.
That's fine. And just following up one of the parts of your talk, Paul, someone was asked, the bit where you explained about the 50%, which are unexplained in that in the pie chart you did, how are you going about looking to understand more about that 50%?
Right. So, I mean, this is it's a difficult question to address. I mean, how do we go about the 50%? Because the reality is that the stuff that is relatively easy to find, we found first.
Yeah.
You know, it's like looking for kind of needles in haystacks where all the needles are a different size, and you know, you can find the big needles very easily or relatively easily, and they get smaller and smaller and they get harder and harder to find. So one of the things that is now becoming possible is that whole genome sequencing is getting to the point where it is almost possible to do whole genome sequencing of thousands of samples. And if we can do whole genome sequencing of thousands of cases and thousands of controls, we hope that we will be able to find a little bit more of that missing 50%. But the reality is that a lot of it is going to be all but impossible to find. They're going to be rare variants with small effects that just, you know, short of sequencing 10 million subjects, which may happen in 50 years' time, but certainly not in the immediate future, we won't be able to get there.
Thanks, Paul. Someone on the chat has asked, what is the survival outcome for patients who are considered cured? So no relapse for 10 plus years after diagnosis of HGSC. I hope that acronym means something to you. And are they really cured? And how likely is it they'll be relapsed after 10, 15 plus years?
So that's a very interesting question. So you have to think about age and stage and BRCA1, BRCA2 mutations as important variables in predicting survival. If you take all women with stage 3 disease, 5-year survival is around 25 to 30%. So in other words, patients still alive at 5 years is around 25 to 30%. And for stage 4 disease, that falls to about 15 to 20%. Now, that does not mean those patients are cured. Long-term survivors, Paul and I have been involved in, one of the things about ovarian cancer is that it's a fantastic community that works together because this is a relatively rare disease. And Paul and I are involved in a large-scale international study trying to understand who are the long-term survivors, who are the people that are cured. What's surprising is there is definitely enrichment for having a faulty BRCA1 or BRCA2 gene because those patients respond better to chemotherapy. And now with PARP therapies, PARP maintenance therapies might actually be cured by maintenance PARP. But if you look before PARP was being used, there are still patients who are cured, and some of them have actually had recurrences and are still alive and still look like they're cured. So we're struggling a bit to really pin down what's different. And I think it's going to come from a combination of the genetic changes in the tumor, BRCA1 particularly, but also the immune responses and the ecology bit about how that individual's cancer interacts with its environment and how the body and the immune system in that individual has fought the cancer. And some of those are going to be determined by the factors that Paul was talking about in his first talk. There are variants that we are inheriting that may make us better or worse at immune responses against cancers. So I don't have an easy answer, but at 10 years, if you've survived 10 years without a recurrence, in my mind, you're absolutely cured, except that we always have to remember that if you've not got a diagnosed faulty gene, you could have a second cancer arising from that faulty gene. So that's relevant in breast cancer and in ovarian cancer. So risk-reducing surgery and other treatments are still important.
You mentioned about how it's a close-knit system that you're working in. Are there any developments in other areas of cancer research that you think will have an impact on ovarian cancer or vice versa?
So I think what the kind of strong thread that runs through bad outcome cancers like ovarian is that they're characterized by this genomic complexity, this genomic chaos, as I called it in the talk, and that evolution and the ability to adapt to treatments is a major reason why treatments fail. Now, we've seen in some cancers with different genomic patterns of change, particularly melanoma and lung cancer, where you get, instead of getting these big cut and paste changes, these copy number changes, you get lots of changes at the base pair level. Immunotherapy is making a dramatic difference in those diseases. And so, for instance, in melanoma, patients are being cured, even though their melanoma has come back by immunotherapy. And in lung cancer, we're seeing definite increases in survival because of immunotherapy helping. So those are important paradigms. They do not, they're actually buck the trend. They don't directly apply to high-grade serous because the wiring diagram is different, and in fact, the trials have been very negative. I think what they force us to do is think about what other cells in that tumor microenvironment and the ecology of those tumors are important. So there are other types of immune cells that we need to target. And that goes back to the previous question about long-term survivors. What else do we need to look at? So we need to learn from other cancers, but I think there are very specific things about high-grade serous that are quite challenging and need to be explored and understood with good research.
Paul, I think you for me.
Well, my interest in research is in genetic epidemiology, and there's lots of developments in gen epi that span lots of different diseases and phenotypes, and we're constantly learning from each other how to do genetic epidemiology better. Something I didn't talk very much about, for example, is polygenic risk scores. The polygenic risk score literature and learning about how to do polygenic risk scores better spans coronary heart disease, psychiatric diseases, and so on. So there are genetic epidemiology is really a very kind of multi-disease discipline in itself. And the other thing about gen epi is that kind of timeline that I talked about is very much dependent on developments in technology. And all of this research, you need the technology to work. And so there's ongoing research that is kind of technological based. And when these technologies come along, whatever, better sequencing, that enables us to do some of the research that we can do better.
Thank you. The hour has flown by, as they always tend to do. So sorry for anyone else who's submitted questions that we won't be able to cover. But just to thank you both, Paul and James, again for your time today, and thanks to everyone who joined today's event. The next talk that we'll be having a break over the summer. The next one is on the 21st of September, looking at brain cancer, and more information will be available on the CBC Eventbrite page, and more information will also be available on social media. So with thanks to everyone again for the time. I wish you all a lovely weekend. Take care.
Thank you very much,
everyone.
Bye.
Bye.